You might be surprised to hear that the Earth is currently experiencing a mass extinction, and it's all because of our actions. More than 20% of species on the planet today are at risk of extinction, and that number could rise to 50% by the end of the century if we don't take action. But what if I told you that extinction isn't so inevitable? Thanks to advances in science, we can even try to turn back the clock by reviving some long-lost species. So, without stalling, let's take a look at some of the extinct animals that could set foot on Earth again. The woolly mammoth, a species of elephant that roamed the Earth during the Pleistocene epoch, commonly known as the Ice Age, disappeared about 4,000 years ago, mostly because of human hunting and changes in climate. Now, researchers at Colossal Laboratories and Biosciences are using CRISPR gene editing techniques to add mammoth DNA to elephant embryos. They want to create a hybrid creature with traits similar to the extinct mammoth. The scientists behind this project hope that a hybrid could help restore ecological balance in regions where elephants are under threat, such as the west coast of Africa, the African savanna, and Southeast Asia. Despite the project's longevity, there are still some significant challenges to overcome, including the complex process of DNA sequencing and the difficult task of obtaining usable mammoth DNA. The steppe bison was a huge woolly cow that once roamed the grasslands of Europe and Asia. It weighed up to 1,984 pounds and had long horns that were 39 inches wide. It came from eastern Eurasia and made its way to North America. The steppe bison was a vital source of sustenance for early humans, and its remains have been discovered at Neanderthal hunting sites. Unfortunately, the steppe bison went extinct approximately 10,000 years ago. However, its legacy endures in the form of the American wood bison. In recent years, scientists have endeavored to resurrect this lost species. Since 2015, South Korean and Russian researchers have been working hard to clone the steppe bison using a Canadian wood bison as a model. After retrieving an ancient steppe bison tail from Siberian permafrost, these scientists are carefully planning to extract DNA from the tail for their cloning efforts. Sometimes cloning projects require the use of surrogate mothers from different species. For instance, an elephant might be used to carry a resurrected mammoth to term. The passenger pigeon was once a common species in North America, but it disappeared in the early 20th century due to hunting and habitat destruction. The last captive passenger pigeon died in 1914 at the Cincinnati Zoo, marking the end of the species. Now, scientists are exploring the use of genome retooling, a form of genetic engineering, to bring back the passenger pigeon. This process involves putting together DNA pieces from preserved specimens to recreate the bird's genetic blueprint. Since there aren't any complete passenger pigeon specimens, researchers are focusing on distinctive traits, like the bird's elongated tail, and combining these pieces with the genetic material of its closest living relative, the common rock pigeon. When we think about animals, scientists are trying to bring back from the dead the dodo, a flightless bird that disappeared in the 17th century due to habitat destruction, invasive species, and dwindling food resources, often comes to mind. The dodo was native to Mauritius in the Indian Ocean, and it's a great example of how human activities have led to the extinction of species. As of 2023, researchers are trying to isolate primordial germ cells, PGCs, the precursors to eggs and sperm, which are essential for the dodo's resurrection. They're using DNA from the Nicobar pigeon, its closest living relative. PGCs can develop into all the cells that form the reproductive cells and their supportive structures in an animal. This big project has already got a lot of money invested in it, $225 million, and we're expecting to see some results soon. The Tasmanian tiger, also known as the thylacine, was a carnivorous marsupial that faced extinction in the early 20th century due to hunting and habitat loss. Its natural habitat was primarily in Australia, specifically on the island of Tasmania, where the last known individual passed away in captivity in 1936. In 1999, Australian scientists started an ambitious project to clone the thylacine using DNA from well-preserved specimens. They managed to replicate 4 complete sections of the tiger's DNA before the project was halted in 2005 due to inadequate facilities and the poor quality of the specimens. Currently, scientists are trying to bring this iconic species back, with the University of Melbourne leading the charge in collaboration with a US biotech company. The Pyrenean Ibex, a subspecies of the Spanish Ibex, was a wild goat that was on the brink of extinction in 2000 due to excessive hunting. The last member of this species died in Spain after being hit by a falling tree. But the Pyrenean Ibex had thrived for millennia across Europe, Asia, and Africa. In an ambitious attempt to revive the species, scientists in 2003 started a cloning project using a method called nuclear transfer. This cloning technique involves taking the nucleus from one cell and putting it into an egg cell that has had its own nucleus removed. The cell then divides and grows into an embryo that is genetically identical to the original donor. Unfortunately, even though they made some progress in the cloning process, the cloned Pyrenean Ibex didn't make it past its first few days. The great auk, a flightless seabird native to the North Atlantic, went extinct in the 19th century due to excessive hunting and egg collection. Recently, scientists have identified the great auk as a potential candidate for de-extinction efforts. They are currently focused on the in silico phase of their research, which involves using computer simulations and bioinformatics tools to analyze and reconstruct the genetic material of extinct species. During this phase, researchers use genetic data from fossils and other sources to put together the complete or partial genome of the long-gone species. The gastric brooding frog, or platypus frog, was an amazing Australian amphibian known for its unique reproductive process. The female would eat her fertilized eggs, keeping them in her stomach until they were ready to hatch. Sadly, this incredible species disappeared in the mid-1980s, possibly due to a fungal infection. In the early 2000s, scientists started looking into the possibility of reviving the gastric brooding frog. In 2013, Mike Archer and his team from the University of New South Wales made a big breakthrough. They cloned embryos of the extinct frog using genetic material preserved in museum specimens. They then placed the embryos into the eggs of a closely related species, the great barred frog. While the embryos didn't reach maturity, this experiment was a big step forward in the pursuit of de-extinction research. The Carolina parakeet, a colorful bird, used to live in more than half of the United States. Unfortunately, they disappeared in the early 1900s. Nobody knows for sure why, but habitat destruction probably played a part. Scientists are interested in learning more about what happened to them so they can help protect birds today. In 2019, researchers made a big breakthrough by sequencing the DNA of the Carolina parakeet. They used the shin bone and toe pads from a specimen that had been hunted, preserved, and mounted in the 1800s. By comparing this DNA with that of its closest living relative, the South American sun parakeet, they wanted to understand what caused the Carolina parakeet to go extinct. Their findings suggest that a poultry disease, likely introduced through human intervention, may have played a significant role. While we don't have all the answers yet, the successful DNA sequencing of this extinct species paves the way for future research and perhaps even the possibility of revival. The Smilodon populator, also known as the Saber-Toothed Cat, was a big cat that roamed North and South America during the Pleistocene epoch, which ended around 10,000 years ago. They're famous for their long canines, and they disappeared along with many other large mammals in the late Pleistocene extinction. Fossil evidence of the saber-toothed cat was found in Brazil in the 1830s. In a big step forward, scientists in 2020 managed to sequence the nuclear genome of this ancient predator, marking one of the most ancient genome drafts in South America's history. Despite this achievement, several challenges emerged. The ancient remains of the saber-toothed cat are severely degraded, making it difficult to revive them. Also, we don't know what the impact of reintroducing such a big predator into modern ecosystems would be. The ecosystems of today are very different from those of the Pleistocene era. The little bush moa, an extinct and tiny member of the moa family, used to thrive in New Zealand. This flightless bird species disappeared in the late 13th century, but there's a glimmer of hope for its return in the labs of Harvard University, where scientists are working on an ambitious project to bring the MOA back to life. The project kicked off in 2018. It involves inserting reconstructed MOA genomes into the eggs of living bird species. The genetic blueprint for this project was put together from the toe bone of a museum-preserved MOA specimen. Researchers are working hard to share their findings in the university's academic journal. At the moment, preliminary results are available on the website, waiting for peer review. The ongoing research is pushing the boundaries of science, with the ultimate goal of achieving de-extinction for the MOA and other lost species. The quagga, a unique subspecies of zebra native to South Africa, disappeared in the late 19th century. It was killed off by hunters who wanted to take its skin as a trophy. The quagga's stripes were only on the front of its body, so its memory is a reminder of the loss of biodiversity. The Quagga Project, which began in 1987, is trying to bring this unique zebra cousin back by breeding them carefully. Eric Harley is in charge of the project, which aims to breed zebras to look as much like the quagga as possible. Some people think this is just a show, but Harley and his team are determined to bring back a piece of the past to the present landscape. The cave lion went extinct around 13,000 years ago, with its demise in Eurasia estimated to be between 14,900 and 14,100 years ago. The Guardian reports that these lions were hunted by both Neanderthals and Homo sapiens. Efforts to bring back the cave lion have been underway since 2017, led by a team of Russian and Korean scientists. They're trying to clone the species using the preserved remains of two Ice Age cubs, Uyan and Dina, that were found in Siberia. One of the cubs' remains had traces of mother's milk in it, which is really helpful for the research. By looking at this milk, scientists hope to figure out what the cave lion ate. Given how well the cubs' remains have been preserved, there's a good chance the species could be recreated, as Dr. Protopopov said. The McClear's rat is a rodent species native to Christmas Island in the Indian Ocean. It was first documented in 1886 and lived in the island's forested areas. This nocturnal creature played a crucial role in the local ecosystem until it became extinct. This was due to the arrival of invasive black rats in the early 20th century. The black rats not only ate the McClear's rats, but also gave them diseases and made them less common, so the last sighting was in 1903. In 2022, Dr. Tom Gilbert from the University of Copenhagen said he wanted to try to bring the Christmas Island rat back. Since the McClear's rat is 95% genetically similar to the Norwegian brown rat, this project is more feasible than reviving ancient species like the woolly mammoth. To bridge the genetic gap, scientists plan to modify the DNA of brown rats, aiming to replicate the missing 5%. While this approach has potential, it's not guaranteed to succeed. This video got pretty long, thanks if you watched it to the end. See you in the next video.